Thank you. I am by the names of Iradukunda Jandamu. I am going to present the important topics related to immunology, and these are the contents we are going to follow. The first topic will cover the risk of chimeric antigen receptor T cell therapy. The second will cover the immunotherapy in the treatment of autoimmune diseases. The third one will be B cell activation by the help of T helper cells. The fourth one will be the regulation of isotype switching. It will be followed by importance of the T follicular helper cells, which is the last one. So let us go one by one. What are the risk factors of chimeric antigen receptor T cell therapy? First of all, this chimeric antigen receptor T cell therapy, it is a form of immunotherapy that uses modified T cells in cancer treatment, and this is done by modifying the receptors found on T cells in order to give these T cells ability to recognize and sensitize the antigens in the cancer cells. So the risk that this current therapy will have, the first one, it is cytokine release syndrome. This normally it is a syndrome which will happen when the immune system become overactivated, leading to the release of large amount of cytokines, which are the signaling protein that regulate immune response. The second point is about neurological toxicity. It is also known as immune effector cell-associated neurotoxicity syndrome, and it is potential side effect of certain immunotherapy, including chimeric antigen receptor T cell therapy. This occurs when the central nervous system is affected by immune response triggered by the third point, it is B cell aplasia. Normally, this B cell aplasia, it is a condition which is characterized by absence or severe depression of B cells, which is the type of WBCs help to produce the antibodies against infections. This chimeric antigen receptor T cell therapy led to B cell aplasia, where it is targeting the antigens found on B cells itself, found attacking and targeting those antigens found on normal B cells, and this will lead to B cell aplasia because the normal B cells have been attacked and targeted or destroyed by this therapy, and there will be no production of antibodies, which will lead to make the person to be susceptible to infection. This B cell aplasia have effect on the immune system. The first one, it is there will be loss of antibody production. Yes, of course, as there is no more B cells to produce antibodies, we will lose the ability of the body to produce the antibodies. This B cell aplasia, it is characterized by recurrent infections, as there is no antibodies to defend the body against infection. The fourth point, it is tumor lysis syndrome, and this is a condition which occurs when large number of cancer cells are broken down or lyse rapidly, leading or making them to release their contents into the bloodstream. The symptoms will be the kidney dysfunction. This is normally due to uric acids. Under the kidney function, we have decreased urine output and even squaring or fluid retention. This is the diagram summarizing the tumor lysis syndrome. As you see, the cancer cells has lysed, and here they have released their content into the bloodstream. This is the blood vessels. The fifth point, it is graft versus host diseases, and normally this happens when the transfused blood which contain the T cells are introduced in the Patients and the donors T cells recognize the recipient cells because they recognize the recipient tissues as the foreign and start attacking and destroying them. The next question or the topic is how the immunotherapy is used to treat autoimmune diseases. Here we have to first understand what is autoimmune therapy, immunotherapy and autoimmune disease. Autoimmune disease are conditions in which the body immune system mistakenly attack its own health tissues and organs. Then immunotherapy, it is a type of medical treatment that harnesses the body's own immune system to fight diseases, particularly cancer, infection, and some autoimmune or inflammatory. Here, these are the examples of some autoimmune diseases, rheumatoid arthritis, and even the first, it is immunosuppressive therapies. Here, these drugs, they are aimed to suppress the overactive of the immune system that is causing tissue damage in autoimmune diseases. The goal is to prevent or to reduce the inflammation, prevent organ damage, and control symptoms. The example which may be used, it is nucleus factor inhibitor. Actually, this blocks the excessive inflammation which may lead to tissue damage. The next point, it is targeted immunomodulation. This approach involves manipulating the immune system's activity to retain or to restore tolerance to the body's own cells. Example, here we use the immune checkpoint inhibitors. Plasmapheresis. The plasmapheresis, it is used to treat some autoimmune diseases like myasthenia gravis and GBS, where we take the normal plasma which contains the normal functional antibodies, and these antibodies are introduced in the body so that they can retain the lost antibodies which are not functioning. The next point, it is intravenous immunoglobulin. This is done where the blood product containing antibodies from the health donors are corrected and introduced into the patient. This also may help to treat the autoimmune diseases. This is done by modulating the immune system to prevent it from attacking the body's own. The other point, it is hematopoietic stem cell transplantation. As we know, these hematopoietic stem cells are the one to produce the B cells. Once it is producing the B cells which are recognizing the body as the foreign, we said, or we find how we can replace these hematopoietic stem cells so that the normal B cells can be produced and produce the antibodies which will be able to fight or to recognize the self from non-self antigens. The next question, it is how T helper cells support B cell activation. T helper cells, it is most potent in T cell activation, where it provides the signals that help B cell to differentiate into plasma cells, and these plasma cells will produce the antibodies. This interaction between T helper cells and B cells is a key part of the immunity. Here are the steps followed to activate B cells by the T helper cells. The first point, it is antigen recognition. This is done where the B cells recognize the antigens first recognize the antigen using its receptors, B cell receptors, internalize them, and this B cell will represent this antigen to measure its compatibility complex class 2, and this complex will be recognized by T helper cells using T cell receptor cells. Upon recognizing or after recognizing this complex, the T helper cells will be activated by producing cytokines. The cytokines especially will be produced by T helper cells from a humoral immune response. These cytokines produced will help in activation and proliferation of B cells, and this will make it produce the antibodies able to fight against the infection. The next point, it is isotype switching and its regulation. The first of all, isotype switching, it is also known as class switching recombination. It is a process in which B cells change the type of antibody they produce without changing the specificity of the mechanism. Isotype switching involves DNA recombination in a constant region of the antibody gene. The constant region of the antibody heavy chain is replaced, guided by switch region in the immunoglobulin gene. Requires activation encoding cytidine deaminase to induce DNA breaks in this region. Regulation. How it is regulated? It is regulated by cytokine, which is secreted by T helper cells, where the interleukin 4 induces switching to immunoglobulin E or immunoglobulin G1. TGF-B induces switching to immunoglobulin A. Interferon gamma induces switching to immunoglobulin 1 in human. CD40, CD41, 40-elo interaction, essential for AD activation and CSLR initiation. Defective isotype switching can lead to immune deficiencies such as hyperimmunoglobulin M syndrome. This is the diagram summarizing how the isotype switching is done and how it is regulated. The next topic, it is how or what are the T follicular helper cells and why they are important. T follicular helper cells are a specialized subset of T cells that are essential for promoting B cell activation, antibody production, and generation of memory B cells. They are critical for forming germinal centers and supporting isotype switching to maturation. Here there is important definition or important point you have to note because it will help us in the previous slides. There is a germinal center, which is specialized microenvironment within secondary lymphoid organs such as lymph nodes, spleen, and tonsils, where B cells undergo critical processes of proliferating, differentiating, and affinity maturation in response to. So we are going to look why the T follicular cells are important. The first of all, it is important because it facilitates the B cell activation and antibody production. These follicular T cells provide a space where B cells undergo full activation, undergo class switching, or produce high affinity antibodies. So without these T follicular helper cells, B cells cannot undergo full activation, class switching, and produce affinity antibodies. Again, these follicular T cells help to enhance the long-term immunity as it helps to produce or in the production of B cells, which are immunological memory. They are necessary for faster and more robust immune response to future infection, providing immunological germinal center formation. As we have seen, this germinal center support the formation of these T follicular helper cells help in support of formation of germinal centers where B cells undergo somatic hypermaturation, affinity maturation to improve antibody specificity. Again, these support immune system adaptation. T4 helper cells help guide B cells to produce different classes of antibodies: IgG for systemic immunity, IgA for mucosal immunity, IgE for parasitic defense, which is crucial for adapting the immune system to various types of pathogens. This is the diagram explaining how T4 helper cells help in the activation of B cells, and these B cells will produce the antibodies which are able to fight against the infection. This is the source of reference that I have used. Thank you for following me. Thank you for watching my video.